Jaenisch. It's a keynote speech. I think I don't need to introduce Rudolf because he's very famous. But if you allow me to say a few words about him, Rudolf is originally from Germany, and he is a pioneer in gene transduction in mice, and he has always been the leader of the field of epigenetic regulation of mammalian development. And Rudolf always incorporates cutting-edge technologies to his research and ask very important questions in biology and medicine. I was surveying Rudolf's papers, and I found he has more than 300 publications, and among them, at least 25 have been cited more than 1,000 times. So you know the impact of his research. And Rudolf is using pluripotent stem cells to understand the genetic and epigenetic basis of development and disease. And I believe he's presenting some of his recent works. So Rudolf, please.
So thank you, Hero, for the introduction, and for the other organizers. For inviting me to this really appearing really great meeting. I'm really looking forward to this. So I would like to talk about epigenetics and some issues of studying it for disease research, and just reminding you, and I don't want to go into this in any detail, but epigenetics really is the way how the environment interacts with the genome. And it obviously has major associations with disease like cancer, neurodegeneration, imprinting, aging, and importantly, for interest for many here, is it's reversible. And for example, in cancer therapy, these are efforts to reverse silencing of some key genes. And of course, epigenetics and methylation really have a major role, the major role in differentiation. So a few questions which I think we as a field need to answer. I think we want to answer in aging and diseases, are there really consistent changes during aging, or is it just random? We want to know whether there's connection to long latency age-related diseases such as Alzheimer's or Parkinson's. Are they similar, different from normal aging? Are there specific loci? Environmental genome interactions, the effect of toxins, of stress factors, of diet. Again, is it specific? And finally, causal relations. Is it causal or is it just associated? So in aging, of course, one big part is epigenetics, to think that plays a major role, but it's really not clear at this point. There's no consistent picture. Does it go up, go down? Probably both. So I think we are really rather not in agreement on this with the data we have. Now, clearly, the exposure to physical exercise, to lifestyle, stress, by chemical issues, diet, we're exposed to this all our life. And we change methylation normally or in disease-related aging, either hypermethylation or hypomethylation. And actually, there is some efforts, Steve Holbert particularly, to correlate the sort of methylation clock with the chronological age. I just took one of his figures where you plot the chronological age against some DNA methylation change, either up or down, and he argues he can determine the age of a person by plus minus 5 years from the methylation in the blood. And of course, all these issues, I mean, your diet probably have change or delay your aging-related methylations or increase it, whatever. So I think there's a very complex interplay which we would like to understand. This study, initiated more than 20 years ago by the NIH, is a religious order study where they got a religious order to donate their brains and their medical history to science. And the first results of this were published a few years ago, where they showed there were, they argue, the authors, there are consistent changes of methylation, for example, in Alzheimer's. The problem here is that cell heterogeneity. So clearly the glial neuron ratio is different in a control versus an Alzheimer patient. So they might look partially just at this. And so that's, I think, is an issue. So when you think about environmental stress situation in disease phenotype, it could be either not related or interrelated or many, many different relations. The issue, I think, is, in contrast to cancer, which is homogeneous and clonal, epigenetic change in response to environmental stress in the brain will be just really very diverse. Every neuron will be affected differently from its neighbor. So biopsies of such tissue, their brain will give quite limited information. It will be just the mean. So the challenges are that we have all risk factors and protective factors genetically, and you're exposed to different stress factors. And in addition, the phenotype might be only relevant for one cell type, but not for the others. So all this asks the question, how do we sort out these genetic versus environmental factors? And we believe, and many do, that we need a single-cell approach to do that. So I'm going to talk about two topics. One is epigenetic regulation in development disease by looking at dynamic detection of methylation at single-cell resolution and editing the methylation from some disease-relevant genes. Now, all of you know this really, how methylation changes dynamically over development, goes down and up again. It's very dynamic. And we would like to understand this. We would like to follow that. And the way it is done, methylation is measured now by whole genome bisulfite sequencing. This is really very accurate based on their resolution, but it is really a snapshot. It's not dynamic. So the current challenges are that when you change from state A to B, there is a lot of gray in between, very heterogeneous. And so what we like to do is to follow this dynamically and to do it at single-cell resolution. So we needed a traceable readout for real-time changes of DNA methylation at single-cell resolution. So the idea for this was that methylation spreads. So if you have some site you want to measure, it gets methylated, the methylation spreads to the adjacent sequences. So if you would insert a reporter here, you might be able to measure the methylation of what you want to measure upstream. And this is really work by Jonathan Stelzer, and I've talked later by work of Sean Liu on methylation editing. And the key was he needed a neutral DNA methylation reporter, not something which changes what you want to measure. And this was really, we came to imprinting, because in imprinting you have these DMRs, which are differentially methylated on the two alleles, and they are maintained, and of course doing primary germ cell development, they are raised. But these are differentiated methylated regions, and the expression of the adjacent allele-specific genes really depends on the methylation state. So this depends on methylation, on DNA methylation promoter activity. It's not regulated in a tissues-dependent manner, and it's ubiquitously expressed. So that was the idea that maybe imprinted genes which contain a DMR in their promoter might be neutral reporters. So for this, we used the SNRP end promoter driving the FP, and just to show that it works, we inserted it close to a GAPDH CpG island, which is unmethylated, or close to DASA CpG island, which is always been methylated in ES cells. And the prediction would be this would be unmethylated and GFP positive, this would be methylated and negative. And indeed, that's what you see. GAPDH insertion, these cells are GFP positive, and both the island and the promoter unmethylated, and insertion downstream of the DASA CpG islands is not expressed, GFP negative, and they're methylated. So this works. And so it's neutral, and we have inserted this into endogenous promoters, in enhancers and super enhancers. It really works very robustly. And we can really use it now dynamically. Look at differentiation, for methylation changes of a single gene, or in reprogramming the reverse. It's very faithful. So I want to give you one more critical example where we put the reporter in the two alleles of an imprinted region, DLK1, DO3, And there's a DMR here, which is differentially methylated, and the maternal allele leads, unmethylation leads to expression of these non-coding RNAs, and methylation in the paternal allele to expression of these proteins. And so we inserted the reporter downstream of this DMR, and the predictions are when you insert it on the maternal allele, the tomato, it should be on, and on the paternal allele in the same nucleus, GFP should be on. Right? So, and that's indeed what you see. Here these ES cells, they are red, but they're not green. So that was fine. But okay, that was expected. But now can you use it? And we were interested really in maintenance of methylation and of imprints. And I said it's stable, and actually it turns out to be not that stable. So we used the maternally transmitted GFP reporter, put it into a blastocyst, made a male chimera, mated this with a normal female, and we get all the offspring of those would be negative because it's a paternally transmitted allele. But in the F2, all the offspring of a daughter should be, sons and daughters should be GFP positive, they stay negative because now it switches to the female transmitted. So that's what the expectation is. And that's exactly what you find. Paternal transmission, no GFP, maternal transmission, GFP in this case in the retina. So it's really the maternally transmitted allele is not, the paternally transmitted allele is methylated. So, and this gives you in the brain, we'll talk a little bit about the brain. So maternally transmitted, the brain is GFP positive, paternally not. And if you now go to the more detailed level, in the cerebellum, for example, and you look at the Purkinje cells, you've also surprised. You see areas in Purkinje cells are all methylated. There's no green. It's like clonal. They're all the neighbors. Or other areas where they're all unmethylated. They're green. So as if there is, that was my initial hope. Maybe this is the memory of some activity these neurons had. Who knows? So anyway, the epigenetic methylation heterogeneity is heterogeneous at the single cell level. And this you don't find only in the brain, but in most tissues, for example, in the gut, in the intestine, you find really the GFP positive cells alternating with methylated cells. And here in the liver, it's sort of very similar. So it's very heterogeneous. So let me come to some conclusions here. So the gain of parent-specific methylation is probably irreversible during development, but the maintenance of imprints is very heterogeneous. And that's, we don't understand quite what the role of this is. We don't know whether that's important, whether it's really functional to have this heterogeneity. The right experiments really have not been done with yet. The question is, effect of agent or environmental effect such as exercise. Would it affect these things? So as I said before, so would you, and you age a mice or you let them run, do they change? Is it really something which reflects the environmental issue? So particularly we were interested in, would this pattern which I showed you before, would this change somehow? Okay? So we let the mice run. And this is Jonathan with his running machine here. This is sort of a wheel where the mice can run, and we can measure how much they run per night, right? So that's this concept. And so the question to the audience is, what's the distance the mouse runs per night? Any suggestions? Well, it's 11 miles. So it's quite a distance for these little beasts. So when you jog in the morning, you should think about this, what these mice do. So we're doing this, and we haven't really seen a major effect. So maybe this is the wrong locus, we don't know, but I will come back to this. But there was something which was very surprising, which I want to bring up, which is sex-specific imprinting, which we did not expect at all. So if you look at the maternally transmitted allele, the offspring, as expected, everything is positive. They're unmethylated. You would expect that. But when you look at the paternally transmitted allele, you would expect everything is negative, methylated. But that's not the case. You probably can see that. You see here in these offspring at different stages of development, the males are positive, they're GFP positive, and the females, the sisters, are always negative. So that's very strange. There's the same allele, the same parental allele. And when you look here, so in maternal transmission, everybody is positive, as you would expect. Paternal transmission, the females, the daughters, behave as you would expect. The males do not. The brothers do not. That demethylate this allele, the paternal allele. So that was quite strange. And so that's really the conclusion. So we asked the question, so this would give you some sorting the muscle cells, and you can see that in a male, you find substantial GFP positive nuclei, and not in the brain of the male, not in the liver of the male, and not in the muscle of the female. And you can stain for this. Males, the muscle stains for GFP, whereas the sisters do not. So it raised the question, is that cell autonomous or non-cell autonomous? So is it the presence of the Y chromosome which makes it, or is it something else? So we asked this question, and one was in vitro. We just differentiated the ES cells to muscle cells, and I don't want to go through this in any detail, but there was no evidence it was sex-specific. So the paternally transmitted allele was always methylated. So clearly either the system was not good enough for us to get a conclusion, or certainly there didn't any readout which was useful. So then we thought we want to do it in vivo. And this is an old mode, the so-called sex-reversed mice. So these are mice which carry a mutation in the SRY gene. The SRY gene is a key gene for sex differentiation in the male. And the XY mutant males are males because they have an SRY wild-type gene on an autosome as a trans gene. So they're males. And you mate those with a normal female, and you get 4 genotypes. You get the same male back, so the XY male, which has a trans gene. You get an XX male. He's male because he has a trans gene. And you get an XY female. This animal is a female because it carries the mutation of SRY but doesn't have a trans gene. And you get normal females. So that's what's really straightforward. So we put then the maternally transmitted allele, paternally transmitted allele, tomato allele into those, and ask the question, who is expressing? And so when you see this here, so these males here, which are males because they have the trans gene, they're red, so they're clearly unmethylated. Those males which don't have a Y, but they're still males, because they have the trans gene, they're red, they express it. Those which have a Y chromosome, but they're females, because they don't have the S or Y functional, they're methylated. And of course, those are also methylated. So what it raises the question is, the effects of sexual dimorphism on parent-specific methylation. Hormones, testosterone, for example, regulate the methylation. We don't know. We haven't seen this in vitro culture, but maybe our cultures are not really good enough to make this muscles. We don't know yet. So it's unresolved at this point what's going on. Okay. So let me switch and come to disease issues. So diet, environment alters epigenetic state of the genome. And as I said, may affect incidence of long latency, late stage diseases. And we're thinking about cancer, pretty well established, and maybe neurodegenerative diseases. So that's really what people think. And the question is really, does diet affect gene expression? Is there concrete evidence for this? And I'll give you one, I think, classical example, which are these 4 mice. They have the same genotype, but they're very different phenotypes. They're slim and brown, they're mottled, and they're yellow, fat, and get tumors. And the reason is, in these pseudo-agouti mice, is that upstream of the agouti gene, there's an insertion of an IAP element. If this element is methylated, nothing changes. If it's unmethylated, it activates this very strong promoter, which now drives ubiquitously agouti expression. That's why the mice are yellow, and that's why the mice get fat and tumors, because this protein interacts with the pituitary. So when you plug this here, the element is unmethylated, you plug it here, it's methylated. So it's really established early development and clonally inherited. We see this as a code color. And this has been used by a number of people to look really at environmental effects. For example, in this from Jaenisch's review, when you expose mice which are pregnant with these genotypes to either high methyl donor diet or low methyl donor diet. High methyl donor diet would be fruit and vegetables, low would be McDonald's. You shift the ratio between yellow and pseudo-agouti mice. So that's interesting because it says that in a short period during development, you expose these fetuses to different environmental conditions, they have long-lasting, stable effects over the lifetime. And also this state is transmitted to the next generation. So a yellow mother has more yellow offspring than an agouti, pseudo-agouti mother. So it's somehow transmitted, not Mendelian, but it ends up in the next generation in terms of affecting the phenotype. So coming back to this, the question we're asking is, what could serve as a sensitive reporter for environmental effects, aging, disease at single-cell resolution? So given that this pseudo-agouti allele is called a metastable allele, it's really sensitive to environmental effects. We're now inserting our reporter into this locus and hope we can now look at any tissue, not only at the code, in single-cell resolution, the brain and any tissue, at changes which occur in aging and in whatever stress situation you want to put these animals into. So this is ongoing. So I said changes in methylation are in principle reversible, and this is really used in cancer therapy. But the current drugs use really epigenetic modifiers, and they're not specific. It's not possible. So you always worry about side effects. And side effects for cancer might be more tolerable than side effects for long treatment, for example, of epigenetic diseases. And so that is an issue. And so we ask the question, can we change a methylation state of specific endogenous loci? And so this was basically editing the ability state without changing the DNA sequence. And this, as you're probably all aware of, is based on the deactivated Cas9, which this mutant doesn't make a double-strand break anymore, and you can fuse to the protein either a TET domain for demethylation or a DNMT3A domain for de novo methylation, which is targeted by a guide RNA to whatever you want to do. And so when you do that, and here will be the two reporters I talked about before, the reporter insertion at the DASL locus, which is all methylated. When you now target TET to it, it becomes demethylated and GFP is turned on. And if you target the GAPDH reporter, which is unmethylated and GFP expressed, we target DNMT3A to this, then it becomes methylated and turns off. So it works quite well. And we can do it also with endogenous genes, like the BDNF promoter, which at the exon 4, there's a promoter which is activity activated. So you activate the neuron, then the promoter gets demethylated and BDNF is expressed. So we can target the TET to this promoter and activate the gene at the same level as activation of the neuron would do by KCR. We can also demethylate a superenhancer upstream of the MyoD gene. And when you do this, you activate MyoD and it leads to myogenic conversion. And also you can block the CTCF mediated looping in the chromatin. So CTCF are really the elements which make these chromatin loops attached. And when you methylate one of those, then you open the loop and you activate the genes in the adjacent loop. And this, for example, occurs also in cancer. It can be methylated in certain cancers and activate some adjacent oncogenes, for example. And in addition, you can do it also in vivo. We can inject these vectors into the brain and affect methylation of the specific locus. So it works quite well. So can we use it for diseases? And the diseases which offer themselves as cancer, imprinting diseases, and autism syndromes. And I will talk about autism. So we'll talk about Fragile X. Fragile X is the most common inherited form of intellectual disability for males. It's X-linked, so females in general are not or very mildly affected, and is caused by the silencing of the Fragile X mental retardation 1, FMR1 gene, which is located here at the end of the X. And it is due to, it's an expansion disease. It's due to the expansion of a short GGC, so VGC repeat, which is 6 to 50 in normal individuals, up to more than 200 in a Fragile X patient. And the result of this is, so the majority are these due to the repeat expansion. The silence promoter shows heterochromatin markers like DNA hypermethylation, histone hyperacetylation, histone K9 methylation, K20 methylation, and so on. So it's typically a really shut down gene locally. So this is the normal situation, and this will be the Fragile X situation. And the question is, when you target now a TET to this repeat, would you affect the promoter, which is methylated in the Fragile X, and affect expression? So the idea would be, so this would be schematically, this is a repeat upstream of the ATG. This is the promoter. Both are methylated in Fragile X. We can target the repeat and ask the question, what does it matter? And later we'll talk about targeting the promoter. And then we can measure this in iPS cells in neurons in the brain. So this is the control cell, 20 repeats. Here's the flanking sequence, and this will be one of these Fragile X patient-derived cells, which has 450 repeats, and they are all methylated. It's a big target. And so the question is, when you now target, we target one guide RNA to this, pretty simple, and ask the question, what would be the effect of this? And the effect on methylation was pretty dramatic. So you can see here that the methylation is totally methylated in the Fragile X cells. When you target a mutant TET, there's no change, but we target the active TET, then it's total demethylation. And maybe more interestingly, the promoter in these patient cells is fully methylated. This will be the control cells, and this would be after targeting TET to it. It basically it's totally demethylated. So the CGG repeat and the FMR promoter are demethylated after targeting the upstream repeat by TET. So what happens to the marks? So this would be just looking at Pol, for example, Pol2 occupancy and some methylation marks. And here's all the three lanes. This is the patient-derived cell. This will be the control, the TET mutant, and this will be the experimental. And you can see you find Pol binding now. HK4 is strong, and HK27, and repressive marks like K9 methylation go down. And as control, the occupancy by of the OCT4 or the MyD promoter is not affected by any of this. So this editing of the repeat activates the FMR1 promoter. So this is really the important result. Expression, does it affect expression? This will be expression in the wild type ES cell. This will be in the Fragile X. This will be again in the TET mutant. And here is the one, the experimental one. So basically, we get wild type level FMR expression. And we see this also on the protein level. This will be the Fragile X ES cells. This will be feeders. They're negative for FMR. This would be after targeting methylation. Again, the control could be positive control, and then the TET. And also by Western, we see FMR being expressed. So that looks all quite good. Of course, if you think about potential therapy, changing this methylation state in ES cells or iPS cells is not very useful. You have to do it in neurons. So we asked the question, can we introduce these vectors into post-mitotic neurons? And so we did this by two vectors have to go into the Cas9, the guide RNA. One of them had a reporter, and then we targeted this. And what we can see is you get about 50% reactivation, is less. And there's also less demethylation. It was only 60%, and the promoter is also not as demethylated as before. We believe this is technical because we only had one reporter to look for we needed double infected cells. We could only look for single infected cells. So it's probably technical. So one key question is, it was all lentiviruses. Are these, no, this was no coming, this was no, can we transplant those into the brain? And would they be stable? So what we did was to treat two, red and the green population of cells. Red were the ones which were edited, green were controlled, put them into the brain of a newborn mouse, and then section it a couple of months later. You can see here is the red cells, which were edited, expressed FMR, whereas the green cells, which were controlled, did not. So this was stable for a couple of months in vivo. Very important was, do we change a cellular phenotype? So we looked at electrophysiology in a microelectrode chamber, where you can put the control and the edited cells in the same chamber. And what is known is that FMR and Fragile X neurons are hyperactive. And what you can see is in differentiation to neurons, you see the control in blue. They have a certain level of activity, but the Fragile X in red have much more. When you target the mutant PET, no change. If you target in green the competent PET, then it goes down to wild type level. So basically, the editing reverses the spontaneous hyperactivity in these neurons. Now, a key question is, how stable is this whole thing? Because it was all antiviruses, which are constitutively expressed. And so we wanted to really remove Cas9. And we tried it with dogs. That didn't work well because the background was so high. So we need to shut off Cas9 totally. And there was this really interesting paper a year ago where this group discovered that bacteriophages produce a peptide which can inhibit Cas9. It's great. This battle between in nature is always so they can just shut off the system which will prevent them to replicate in the bacteria. So we used this ACR2A4 protein to shut off Cas9. And so this is the experiment. Basically, we had to put the two vectors into the Fragile X cells, sort for the double infected cells, then be infected with this construct, sorted for gain transduced cells, and then different times afterwards look for expression of FMR. So this would be with the positive control, the negative control, Fragile X, and this would be now 0 days and in later days expression. And basically we find high expression, and we did it up to 21 days. So you demethylate this and it's stable, even if there's no Cas9 anymore to maintain it. So that was certainly encouraging. So then comes, of course, now a problem. It's clear that GCC is not a rare sequence. And so computationally, there should be about 1,000 foci where this guide RNA should bind. So we asked the question by chip, where does Cas9 bind? And we did this with different concentrations of Cas9. And depending on the concentration, we find on this Manhattan lot between 1650 and 450 binding sites. So that was a bit of concern. And so we asked the question, what's the effect on methylation by sequencing, whatever came down by chip? And this is the result. This is sort of a little bit complicated figure where you plot here the experimental PET against the mutant PET, and we find most of the, in blue color, nothing changes. Many of those, of course, have low methylation, but even some are high methylation don't change. But there are 29 sites which have more than 10% methylation changes. This would be FMR, which is 100% demethylated, but these others, quite substantially, get demethylate, and the blue rings indicate they have a promoter. So what happens to this? So first, POC2 occupancy in K4 methylation. So when you look at the red, at these promoters, they really don't change much. Maybe one exception a little bit here in their Pol2 occupancy, and K4 methylation also. It's really not very much change. It's minimal on these off-target loci. When you look at expression of these 29 sites, they really don't change much, or less than fourfold. And you should remind, these are methylated loci, which are probably off. So you go fourfold above a very low, maybe zero level. So the 29 said the 10% methylation changes showed no fourfold expression change. When you plot this against the methylation, the expression against the methylation, really on a linear plot, you don't see it. It's too small. Here's FMR, it's 800-fold up. But if you go on a logarithmic plot, there clearly you see these few genes which go fourfold up. So the demethylation of these loci has only a small effect on the expression level. Okay, so then you come to summarizing where we are. So targeting this repeat really induces demethylation of the repeat and the promoter. It reactivates the gene in the S cells and postgraduate neurons. It sustains expression after transient editing and in vitro after transplantation into the mouse brain. It restores normal electrophysiology in neurons. So clearly one firm conclusion we can do, demethylation of the repeat is absolutely sufficient to turn on the gene. Unresolved issues are off-target Cas9. Clearly there's DNA methylation changes. There's some gene expression change. Can we prevent this, for example, by targeting the promoter, as I mentioned before? So coming back to what I showed before, we targeted this repeat and we led to demethylation of both. Can you target the promoter? And we're doing this, but it's really clearly, it's not easy. So when we target the 6 or 8 guides, there's no demethylation of the promoter. So what's interesting, the first time it failed. So for us, so the presence of this block of methylation clearly affects what we can do at this point here. So we don't understand the rules. So do we have to target something else like T300 or editing the chromatin? We don't know. Thank you. Okay, so it's my last slide. So just to remind you, methylation changes are sort of well defined in young cells. CpG islands are unmethylated, repeats are methylated, and this change is during aging in some way, maybe even more pronounced in cancer. And you would like to understand this. Are there, as I said before, are there specificities? And so the tools, which I talked about, that really allows dynamic real-time tracing of methylation change at single-cell resolution, and the editing, I think is very robust. And I showed you the FMR, and we're interested also to activate the MECP2 gene, which is cause of Rett syndrome, on the inactive X. So those are issues which we like to do. So let me come to the end. I talked with mostly the work of Sean Liu and Jonathan Stelzer, presented together with many contributions from Marie Fritsch, Yuelin Song, and Stella Marculati. Collaborate a lot with Rick Young. The cells, the Fragile X cells came from Vincent Benisti's lab, and also Hao Wu and John Gray from Qualcomm also contributed. Thank you.
Dear Professor, very nice presentation, and I have 2 questions. And the first one is, I wonder if the methylation can affect the normal DNA replication ability. Because I know that I noticed that you see that fragile X syndrome are caused by the abnormal repeats of CGG. And you know that HD, Huntington's disease, are also caused by the abnormal repeats of CAG. So there are similarity between the 2 diseases because they are both begin with C and end with G. So we can also related to the CG island. So I wonder if the methylation of the CG and G can affect the ability of the DNA replication. And the second one is that since these years the epigenetics has advanced so much, so I think that we should modify our concept of transgene. We should include trans epigene. I want to know how you think of this question.
So I'm not sure if I understood all the questions. The first one was about replication timing. Does replication timing changes? Is that what did I understand this correctly?
You know that the Fragile X syndrome are caused by the CGG repeat abnormal CGG repeat, and HD Huntington disease are caused by the CAG repeat. So there are.
Huh? Oh, Huntington's disease. So Huntington's disease is not a methylation. That's a CGA repeat. It's not methylated. That's something else. So that's toxic. You get this polyamine, or so you get these precipitations. In Fragile X, in the full disease, it's totally shut off. So there's no toxic peptide accumulating. So Huntington's is totally different. That is not an epigenetic disease. It's genetic. And the first question was replication timing. Maybe I missed that. I'm not sure what the question was. What was your
The second one?
Well, maybe you should do them later then. So I missed.
Okay.
Yeah.
It's enough.
Hi, Professor. Hi, Rob. Thank you for the talk. It's interesting about the imprinting gene local reactivation in sex-dependent mechanism. I'm wondering if you do the camera experiment to inject some light, for example, to see the female cell can be reactive in a male camera mice.
So that's all done in human cells, right?
Okay.
So when there were female cells, we put them in the mouse. And in this case, we didn't care, but I don't know even whether it was male or female. We just wanted to see how stable is it, the expression, but we didn't want to see any phenotype or any interaction with the neighboring cells.
But you use the sex reversed model to say XY genotype mice can not reactive the imprinting locals when they are sex reversed.
Oh, you talk about the sex reversal experiment. Okay. No, this was not chimeric. These were not chimeric.
Yeah, I know this is not chimeric. I'm just wondering if you can...
this is something we should do. I agree with you. So then you put these cells, you make a chimera. I think that's a good experiment. We haven't done that. Because they put you in a male environment, one of these cells, you can look at these allele. Would it be activated? I agree. So that's we haven't done.
Professor, so what is the function of the promoter demethylation? Because in your FMR1 model, that you show that demethylation does not affect the PO2 occupancy. So I wonder what's the function?
No, the promoter gets demethylated when you target the repeat. When we target, it's always methylated. It's all. When we target the promoter itself with the guide RNA, he could not be methylated. So there was no effect.
Okay.
So it was just not successful.
So Rudy, I'm interested in the sex-specific demethylation or heterogeneity experiment. So have you tried to artificially manipulate the hormone level, let's say, for example, because it's male, so for example, like a castration experiment, etc.
Right, we have not done this. I think these are all experiments we need to do because we had hoped in vitro would give us the answer, plus minus testosterone. It didn't work, so we couldn't see anything. So maybe it's insufficient, not a good system to do that. So these are experiments we should do, and we're planning to do those. Yeah, that's very good.
Any other questions?
Thank you for your speech. I got a question about the last part. FX activation by Cas9 therapy, but instead of demethylation therapy, does that implicate that the silencing epigenetic state is caused by the expansion of DNA replication, the CGG replication expansion?
You were asking why does this, or how does
The therapy result.
The therapy.
Yeah,
well,
yeah.
The therapy, if you want to do this in a patient, I think you have two issues. One, you have to demethylate this, and the other one, how to deliver, the gene delivery. This is the biggest issue of any gene therapy. And that's, of course, is a totally different problem. So you presumably have to deliver this to quite a large fraction of cells to affect it. So this is something which hasn't been explored at this point.
Thank you. I will think about it later.
I just have a question. So have you tried the anti-CRISPR experiments in ES cells? So the prediction would be that it won't be stable because the ES cells will remethylate the region. Is that right?
You mean the turning off Cas9?
Turning off Cas9 and
this
then...
was done in ES cells. It was done in
that
ES.
was done in ES.
Was done in ES cells.
So then what? Then
the more important is the neurons. We haven't done this, but I assume it will be also stable there, but we haven't done this at this point.
Okay, then during the normal development, what stage those repeats get?
Okay, so this is known from, that's an interesting question. So when you isolate ES cells from an embryo which has the expansion, it's unmethylated. And when you differentiate these cells, it becomes methylated. When you reprogram a somatic cell from a patient which has a methylated expansion, it remains methylated. So clearly when these alleles go through the germline, they're treated differently as when you reprogram them to an iPS cell. And so that's, the methylation is very clear experiments from this and these laboratories.
Okay, there. So thank you for talks. I'm interested in the repeat in human promoter region. So how does CZZ repeat increase? So what is the cause of duplication of CZZ repeat?
Right. So I think these repeat areas are always very vulnerable to mitotic recombination and expansion. So I think that's known from many others. So that's a vulnerability, which with a certain frequency occurs.
Thank you.
Okay, this will be the last one.
Okay, thank you. I have a question. I was thinking about that whether the DNA methylation will cause the repeated expansion to grow its length. Because I read a paper that DNA repeats located in the test bed boundary are more likely to increase their length. So will DNA methylation also have this role? So if it has, so it will cause a feedback loop. It will cause disease much worse.
So are you asking when it's methylated, it will be more vulnerable to expansion than it's non-methylated?
Yes.
Well, this is progressive. So if you less than 200 expand, in general, this is not methylated.
Yeah.
And then it makes actually potentially a toxic peptide from this RNA.
Yeah.
So that has been shown. So that's really not totally clear how that works. But the pre-expansion is a different disease.
Okay.
So it's a different disease than the full expansion. And it's very different molecular basis. One is methylated, the other one is not. One makes an RNA, the other one not.
Yeah, okay. Thank you.
Mm-hmm.
Okay, so thank you very much, Rudolf. Please give big hands to...
So our next speaker is Xiaoyi Wang. We actually intend